Hello everyone. So today we're going to talk about the second part of chapter 19, which is going to be focused on the genetics behind the sexual reproduction that we talked about last week. And we're going to kind of talk about the history of it, how it got understood, how it was discovered, how it was understood through very simple experiments, but very detailed and very carefully done experiments through of Mendel's in plants. And so let's get started. So, as we talked about it a little bit last time as well, Mendel's experiment with peas were what helped us understand the basis of inheritance. And he was lucky, so he was a monk. He worked in a monastery. Obviously, he was in a monastery. And part of what he did, he was very interested in agriculture and growing plants of various types. And some of the things that he noticed was that sometimes he had plants that were always giving rise to the same thing. There was no variability. And other times he was able to see new phenotypes, new things appear from the same plants. So maybe he had colored flowers, and then in there, a subset of them would be white flowers. And he figured that that was interesting. He wanted to learn more about it. So he was able to identify 7 traits where he had what he called true breeding lines of very concrete phenotypes or physical features that he could monitor. And he started looking at them and through very controlled experimentation where he crossed true breeding lines of one trait with the other and saw what he came up with in the resulting progeny. So there were 7 traits that he focused on. Those were pea shape. These are pea plants. Pea shape, pea color, flower position, the shape of the pods that the peas were in, the pod color itself, and then whether the plants were tall or short. And so that's what he started with. Now, before we can talk about what it is that he did and what he found, there are a few terminology related things that we should be aware of. So, as we know, we've talked about this before, last time as well, in most diploid organisms, there are 2 pairs, there's 1 pair of the 2 chromosomes of each type, or a pair of chromosome of each type, 1 inherited from each parent. So these are called homologous or homologs. So a homozygous is if an individual is homozygous for a gene, it means that both alleles of that particular gene are identical. So they are exactly the same, blue eyes on both parts, yellow peas on both parts, axial flowers on both ones, same exact thing. If it's a heterozygous, that means that the alleles that you got from the 2 parents are different from each other. So that would, an example of that would be obviously one parent gave the yellow peas while the other one has the allele for green peas. So now you have one of each, 1 copy of each. And hemizygous is another term that you can use. And hemizygous usually talks about a gene where there's only 1 copy of that gene available. Could be for a variety of reasons. An example would be a gene that is present on chromosome X in a male. So because there's only 1 copy of chromosome X in a male, the other one is a Y chromosome making them a male, then all the genes that are on that chromosome X are going to be hemizygous because they'll only have 1 copy of that gene present. Another might be a situation where maybe that particular gene on the other allele, gene position is mutated or deleted because of some reason. So there's only 1 functional copy of the gene available. Now, where the genes lie, the specific position along a chromosome where a particular gene is called a locus. So each point where there is a gene, that's a locus for that gene. And each allelic form is going to be responsible for that visible trait that you see. So when you have a heterozygous trait, where you have a heterozygous gene, where you have one of the trait on each one of the alleles, then the resulting physical features may be one or the other, or there may be a combination, and that would be dependent upon the type of alleles that you have. So what do I mean when okay, so here let's talk about what some of the other terminology is that we need to know in order to understand Mendel's experiment and to truly appreciate what they mean. So first of all, we need to understand the difference between genotype and phenotype. Genotype refers to basically differences in DNA sequence, so they could be any number of mutations that are found, any number of changes in the gene sequence that are found within a population. All those differences in the gene sequence that you have, all the different flavors of the genes that you have, they are called the genotype. So you usually have a lot more genotypes present in a population for a given gene than what you can observe as outwardly features. And those outwardly observable traits are termed as phenotype. So the phenotype is what you're looking at, and it is usually because of the presence or absence of a gene or presence of the alternate forms of the gene that are available. Those many, many different genotypes may be giving rise to slightly different or incredibly different phenotypic features related to that particular trait. Now, any given population you look at, for every trait that you observe, you're going to see that some traits are very, very, very common, are present in a lot higher abundance than others, and others are very rare. Typically, the most common form of allele in a given population for any observable trait is what we call the wild type form. In addition to that, you may have within that wild type form, if you were to look at the genotypic sequence, you will still see variability within that genotypic sequence. And you will see that there are changes. There are, at sequence level, there are some differences. There are some here and there nucleotide differences or mutations, if you want to call them that. However, they're indistinguishable in appearance at the end of the day, and they all exhibit the same normal or wild type form of the phenotype, and that's why they would be indistinguishable from those wild type individuals. So all those DNA, slightly different DNA sequences, alleles are different at the genotypic level, but yet they're still giving you that wild type form at the end of the day. Another types of mutants, another category of mutants you will see in any given gene, any given observable trait, are alleles with DNA sequences that end up changing the phenotypic form. So changing what you observe at the end of the day for that trait. And now you have a different appearance than wild type. So let's say your wild type form for a particular flower color was pink, but then when you look at the variants, you saw some DNA sequences that still give rise to pink, but then when you looked at the other range of mutations, you saw purple and white and yellow and green or blue or whatever. All of those colors that you're seeing in different forms because of DNA sequence changes are mutants. And all these different mutant alleles of a gene that give you different range of phenotypes, different browns or different colors of flowers, whatever it is that you're looking at, are called an allelic series. So basically a catalog of every single possible DNA sequence change that can lead to a phenotypic change. All those alleles are termed allelic series. So in my case, that particular flower color gene will have in its allelic series all the colors that we talked about, from white to purple to pink, blue, and whatever, green, I think I said. All those colors would be part of that allelic series. And many of these will be a result of usually a single point mutation somewhere along the gene. Sometimes it's more than one mutation, or sometimes it's a little bit more intense change, but majority of the time it's just a single mutation. Now, when Mendel started to look at these types of experiments, what I had the term that I used before is that what he discovered was that there were some true breeding populations in his plants, these pea plants. By true breeding, it basically means that you have an inbred population of an animal or a plant in which all parents and any offspring that comes from the crosses between them always give rise to the same phenotype with respect to that one observable trait. It doesn't mean that these are clones and that they will always look the same and that they will be exactly the same for every single trait. But if I was observing flower color and I was looking at purple flowers and I crossed a purple plant with a purple plant, I would always get purple plants. And that would mean that what I'm looking at is a true breeding line for purple colored flowers. Not for pea color, not for pod shape, not for flower location, but just the color of the flower. Another thing that we do in order to ensure that sometimes that we have a true breeding plant is we take what we call monohybrid crosses. A monohybrid cross is going to be a cross between two individuals that are thought to be or that are heterozygous or a hybrid for a single trait. So in this particular example, we have a true breeding purple flower and we cross it with a true breeding white flower. And when we cross two true breeding lines together with different phenotypes, in my F1, all the progeny, all the offspring are what we would call heterozygous because all of them would have one of the purple allele and one of the whatever allele is making them white. And so they will have two different alleles for that flower color locus. This is called the first filial generation right here. This is your parental generation. Now here in the first filial generation, if we cross these two together, two offsprings together, that's a monohybrid cross. This first one was the parental cross. This first filial cross, in this case, is a monohybrid cross. A single trait, hybrid, meaning two different alleles. And then you will end up with, in your second progeny, different options, and that would be second filial generation. So Mendel's experiment used true breeding varieties, and then he cross-fertilized them with each other and produced hybrid offsprings, which resemble either one or the other parent. So in this case, right here, you have true breeding yellow peas and true breeding green pea plants. And when you cross-fertilize them, you end up with offspring in the first filial generation, which have 100% yellow pea plants. So in this case, the yellow is masking the green phenotype because obviously you still have the green allele in them, but that's not what's showing up. In this case, what we call this is that it is a complete dominance of the yellow over the green. This is where you have the effect of one allele in a heterozygous genotype completely mask the other. So in the above example, the yellow pea color completely masks the green color that's in there at all. The allele that is masking is said to be dominant over the one that it is masking, and that is the recessive form of that particular gene. Dominant alleles in genetics are written with capital letters and recessive alleles with lowercase letters. And then we can also use sometimes superscripts and subscripts to use for different alleles. For your wild type allele, we typically use a plus sign to indicate that this is the wild type version of that allele or the most common version in a given natural population. So here in this example, purple is dominant, white is recessive, and we know that because you have purple flowers that are homozygous and white flowers that are homozygous, but when you have a heterozygous individual, you get a purple flower. and that's how you would know which one is dominant. We can calculate the incidence of each one and the probability that any one of these will appear in a given cross using what we call Punnett squares. In a Punnett square, you write the alleles from each parent, the ones from usually the male on the top and the female on the side, and then you can fill this up and simply by going into each one of these blocks and writing the top and the side side by side. So in this one, it's capital A and little a. So it's a heterozygous, 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 heterozygous. 100% of the progeny from a cross between a dominant and a recessive true breeding plant would give you a heterozygous individual. All of them purple flowers. So that one is your F1, or first filial generation. In an F2 now you have two parents that are both heterozygous, your monohybrid cross. So looking at the progeny from here, it's going to be a little bit different. The parent, this type of cross will give rise to one of the four that's going to be just homozygous dominant, two that are going to be like the parents heterozygous, and then one that is homozygous recessive. However, phenotypically, looking at the phenotype, observable trait, you will find that you have 3-to-1 ratio, 3 with the dominant phenotype and only 1 with the recessive white plant. But now you have the white plant back. So genotypic, the ratio is 1-to-2-to-1, and phenotypic, it is 3-to-1. A test cross is a way to explore if what you're starting out with is a true breeding or a recessive, true breeding or a heterozygous individual. In this case, we're looking at the genotype of the individual to try to figure out what it is. It is, a long time ago it was used, and it's an experimental mating test to see if we could find alleles of different types present inside what looked like a dominant individual. It can help us determine whether our dominant phenotype is homozygous or heterozygous for a particular allele in a given individual. So here we have an unknown dominant phenotype. So it's a black sheep. We don't know if it's a homozygous or a heterozygous black. We cross it with a known true breeding recessive colored sheep. In this case, it is a white sheep. And when we cross it out and we look at the phenotypic ratio, if our unknown is homozygous, every one of the progeny should be black sheep. However, if our unknown is heterozygous, then you have the possibility of getting white sheep as well. Actually, 50% of the individuals would be black sheep and 50% would be white. So you would know within a generation whether it was a true breeding or not, or whether it was a dominant containing heterozygous, containing individual or not. So there are other things to consider in this type of analysis as well. One of them is looking at the F2 generation, right? When you don't have a test cross that you're doing, you have to wait for 2 generations to see whether what you are ending up with is a true breeding or a recessive or a heterozygous line. Because if you had a true breeding line in the first generation, you would, like we said, get all yellow plants, and then in the F2 generation you would see that 3-to-1 ratio. Here is another example where you have your spelled out. This is again looking at the yellow and green peas. In the first generation you get all yellow peas, and then when they self-fertilize you get that 3-to-1 ratio. Parent plants produce gametes in this case, just like you would see gametes in any other organism as well, whether it is humans or another animal or another sexually producing plant. And those gametes will contain only one of the two possibilities that are present in that particular individual for that allele, for that gene. And the phenotype of the offspring is going to be dependent upon what combination they end up with. So, looking at all of this combined, where I went. So looking at all that data combined, Mendel came up with two specific laws. The first one was the law of segregation, which applies to all sexually reproducing organisms, and basically talks about how these genes are segregating on a specific locus. They are going to segregate regardless of what it is that that trait is. A single growth factor gene, for example, has been linked to body size. So if you have just one gene that is affecting body size, it's going to segregate in the same manner. And if you have three, each one of them is going to be independent of each other. So recessive alleles will follow the same Mendelian law of inheritance. So just because you have a recessive allele doesn't mean that it's not going to segregate the same way. So here we are looking at a human pedigree of sorts, and we are looking at how you're inheriting type 2 albinism. Albinism is recessive, so here you have an albino man and then a normally pigmented woman. In the first generation, all offsprings are normal looking, but in the second generation, if you have two individuals mate that were both heterozygous for albinism, you would get that 3-to-1 ratio again for that single gene associated with albinism. So because there's only one gene associating with this, it's going to show this typical Mendelian inheritance pattern. This type of segregation of alleles, this type of inheritance makes it important to increase variability in a population through marriage of individuals that are not closely related. The more closely related two individuals are that are coming into, that are breeding together or that are marrying together, the more likely you have the chance of showing up those recessive, typically detrimental traits for that given population, and thus giving rise to diseases or giving rise to issues that were masked otherwise because of and their recessive nature. So here is an example of how first cousin marriages can increase the likelihood of various recessive mutations leading to disorders, just like deafness, which is a recessive trait. If you have a lot of first cousin marriages, you're more and more likely to see that trait happen, because you have a bunch of individuals in that pedigree already that are going to be heterozygous for that trait, but because they had a normal gene also, a normal allele of that gene also available, they were able to hear perfectly fine. The more individuals that mate like that, the more likely you have these individuals pop up that are going to end up with that trait actually showing up. This is easily seen also if we look across the globe at prevalence of consanguineous marriages. So these are just close first cousin, second cousin marriages, and look at also at the same time in those areas the prevalence of incidence of birth defects. We notice that areas that have high rates of consanguineous marriages, right here in this, upper part of Africa, Middle East, and some of that Asian countries, will also have higher incidence of birth defects in those populations compared to those where you don't see these types of marriages happening. Now, a second type of cross that we want to be aware of is a little bit now more involved. It's looking at two traits. It's called a dihybrid cross. Here you have two different traits that are that we're examining at the same time, and we are looking at how these alleles can segregate independently. So remember, Mendelian law of segregation pretty much says that every allele is going to segregate independently of others. So each allele on its own has a chance to segregate the same way. So those two alleles, they are equal chance that you're going to get a gamete out of it, and equal chance that they're going to get inherited to the next generation. So your final combination is a combination of, final phenotype is going to be a combination of those different various alleles that are present, or different gametes that are present. Now, when you have two different traits that you're looking at again together, each one of them will segregate the same way. So now you have true breeding lines in this case for two different traits. These are for yellow round phenotype. Both dominant traits don't have to be. One of them could have been recessive. And two recessive traits, in this case, green and wrinkled. So these will just give rise to, because both alleles are identical in this case, so you're gonna give rise to all the gametes are going to be dominant for both in one, and dominant and recessive for both in the second. You cross-fertilize, same as before, your F1 generation is all gonna look dominant phenotype, yellow and round. However, this time there are 4 different possibilities for the gametes that form from this cross. And those possibilities would be a combination of these two different genes. So you can have both dominant alleles together, both dominant genes, alleles of the two genes together. You could have both of them recessive, just like the original parents. You could also have one of the dominant type and the other of the recessive type. So you have 4 different gametes that can form in the female and the male, or in the two plants. And looking at the combinations this time, you can give rise to 16 different genotypic, combinations in a way. Some of them are going to be the same, right? But this is going to, in this case, the F2 generation, the second filial generation, will give rise to a phenotypic ratio of 9 to 3 to 3 to 1. Where the majority of them are going to look like the parents, and then you'll have 3 that are completely dominant and 3 completely recessive, 3 that have 1 dominant and the second recessive of each type, and 1 that's completely recessive for both. So important to know that in looking at something like the more genes you have, the more variability you're going to be introducing. Now, where does this happen? It happens obviously at the meiosis level, as the gametes are forming, as in the beginning of the first meiotic division, when the homologous chromosome pairs come together and the homologs have those chiasmata where they are swapping material with each other. You're going to end up with the various varieties of gametes that we just talked about, so that the final 4 that form are going to look very different. So the separation of these duplicated homologous chromosomes during meiosis will be where Mendel's law of segregation and independent assortment takes place. Each one of them, because they're in this case are shown on a very completely different chromosome, they're far away from each other, will assort completely independent of the other and segregate according to its own combination that is present. So basically, they didn't have to be on different chromosomes. They could have been on the same chromosome as well, but just far away from each other. So on average, if you have 2 genes that are far away from each other on the same chromosome, there's very little likelihood that they would both cross over together. And on average, these will then give rise, each one of them will segregate independent of the other. However, sometimes you have genes that are really close to each other that you're examining, right? For example, right here, you have A and B. These 2 genes are very, very close to each other. So it's very unlikely that there's going to be some type of a crossover event just between these 2 genes. And in that case, those two genes may not follow Mendelian law of segregation and independent assortment because they are very likely to cross over together since they are due to their close proximity. This is called linkage between two genes. The closer two genes are to each other on the chromosome or on the arm of the chromosome where they are located, the more likely they are linked and the less likely they are to assort independently of each other and segregate according to Mendelian genetics. But two genes that are far away, for example, A or B compared to F or E, there could be several crossovers happening all along that arm, and each one of them would just move independent of each other. Okay, so now let's move on and think about how the different alleles are coming up. What is causing those different alleles to happen? Well, we already said that it's usually mutations that lead to those changes. And these type of DNA damaging mutations happen because of exposure to DNA damaging agents in many cases, in addition to just like, by chance as the DNA polymerase is replicating and things are happening, maybe a mistake happens that is disregarded and wasn't picked up by the system. But your normal gene sequence, if treated with a DNA damaging agent or a mutagen due to exposure to UV light, due to exposure or ingestion of certain toxins like certain pesticides that are mutagenic, you could end up with higher rate of mutations or end up with mutations that can lead to downstream effect. There are several different ways that genes can get mutated specifically. Some of them is just nucleotide substitution, where in this case a G gets converted into a C, right? So you have a cytosine instead of the usual guanine. You could also have an insertion of an extra nucleotide. This typically happens when you have a lot of repetitive or repetitive-like sequences. So in this case, you have an addition of an adenosine in the sequence, so your sequence got longer because of that. Similarly, you could also have a deletion of a nucleotide as well. Again, both insertion and deletion are more likely in repetitive sequences. So in this case, the G is deleted from the sequence, and then obviously you can have multiple nucleotides added or deleted from a sequence as well. Any one of these cases would lead to a sequence change that can lead to either no change at the end of the day or some dramatic change and the end product, whether it is a protein or a RNA. So mutations in protein coding area of the genes or in the protein coding genes essentially can affect your final protein product. The best case scenario is that it has absolutely no effect. Whatever the change was, it led to the same amino acid being placed in there or an amino acid with the same properties, so a positive for a positively charged one, basic for a basic one, acidic for an acidic one. In that case, you still get a normal wild type protein, the normal wild type phenotype, no change. Other times you have what we call loss of function mutations, where the mutation happened in a way that either made the protein unstable, so it was just never made, or if made, was destroyed. It could be truncated because it converted one of the codons into a stop codon, so the protein transcript ended prematurely and was missing some important catalytic domain. Or it could have a point mutation, just a single mutation, just a single change, but now your resulting protein can no longer bind to its binding partner and can no longer have its effect. All of those are what we call loss of function mutations. Similarly, your protein at the end of the day could also have a mutation that can enhance its function. So that's either more stable and can last longer, can have stronger catalytic activity, or additional binding partners so that it can have additional controls and additional jobs that it can do now that it couldn't do before. So all those would be possibilities when mutations occur. Again, we can look at our phenotypes and we can look at what type of mutations occurred and what kind of downstream effect they presented. Now, many times when we're looking at phenotypes of particular type, one of the things that we look at is, well, is it because of a gene, obviously with different alleles, or is it because of a trait, or whether this trait is not controlled by just one gene, but multiple genes? So many of our genes, many of our traits, observable traits, are actually complex traits. They are not controlled just by a single gene, but multiple genes, sometimes on multiple chromosomes, not even the same chromosomes. So that type of behavior is what we call complementation, where there are two or more genes working together to give you that final phenotype that you're observing. It could be eye color, it could be height, it could be shape, it could be any of those phenotypes. But in those cases, those phenotypes are not controlled just by a single gene. So they're not as simple as just yellow versus green pea allele on a single chromosome, on a single locus, but rather on multiple loci for multiple things. And so we can test if a trait that we are observing is from one gene, is as a result of one gene with two different alleles, or whether it is two mutations in two different genes. And this process is called complementation test. In a simple complementation test, you're starting out with an individual who is homozygous for a mutation that we are examining, that we care about, and we mate it for an individual who is also homozygous for a different mutation, leading to the same type of result, and you look at the final offspring for the answer. If the mutation occurred because of two different genes, then when you cross two different individuals, both with the same mutated form, but as a result of mutations in different genes, when you combine them, the progeny is going to show completely normal phenotype, because one of the mutant will provide the wild type copy of the second and the other the wild type copy of the first, and now your resulting progeny is going to have wild type anomaly at the end, a wild type phenotypic expression at the other end of the day, because they'll be heterozygous for both. In a non-complementation, your two different individuals that were mutated both had mutation of the same gene but at different points. And in this case, crossing them together would still give rise to a mutated phenotype. And you would know that there was only one gene involved in these two cases. Both of them had mutation in the same exact gene, so that even though you combine them together, you still ended up with mutated phenotype. So there were no normal copies of that mutated gene present in that case. We can create, so many times when we are looking at these traits, you have not just two observable phenotypes, but lots of observable phenotypes, especially in most multicellular organisms. So we can create a chart just like this, whereby we can analyze the complementation. It's called complementation analysis chart, and we can utilize this to figure out how many genes are involved in that particular observable trait, and which mutations complement each other, which ones don't, and which one of these multiple range of phenotypes that we are observing are coming from mutations from each one of those genes. Those would be what we call a complementation group. So in this chart, I have all the possible observable traits on the top and on the side, and I essentially create a cross between all of these possibilities. So white with white, garnet with garnet, ruby with ruby, then white with garnet, white with ruby, white with vermilion. Same thing with every single one of them. And then I look at the progeny and I see if there's complementation happening or not. In each of these cases, you wouldn't expect any complementation to occur when you cross them with themselves, right? Because obviously that's the exact same mutation, exact same gene. It shouldn't complement. You should still get the same phenotype, and you do. So in all of those would be negatives. When you see a plus, that means that there was complementation and they were able to retrieve the wild type phenotype between the cross, and that would put them in different complementation groups. So white and garnet are in two different complementation groups. White and ruby also complement and are in two different complementation groups. Now we want to know if garnet and ruby can complement each other, because if they do, then you know that they are also in two different complementation groups. So let's see, there should be a little pen or something maybe. So white is in one complementation group, garnet and ruby, and we know that garnet and ruby complement each other, right? Garnet with ruby complements each other. That means that garnet and ruby are in two different genes, right? So we continue this story and we see which ones are together. So vermilion, again, it's complementing with all three of them. So guess what? It's going to be another complementation group. Now, after that, when we look at the next one, white does not complement cherry, coral, apricot, or buff. That means all of those, none of them are going to be in the same complementation group. Those are all mutated in the same gene, right? C, A, B, and then you will have the carnation separated. And so you will keep going and you would put them together to see how many complementation groups we have and how many different genes are affected. So we see A and B, all of the ones that don't complement each other are going to be mutations in the same gene. And then you go on and you say, okay, can cherry and coral complement each other, right? So you go and see cherry and coral, they don't. So cherry and coral are in the same complementation group. Then you go and see, well, do cherry and apricot? They don't. Cherry and buff, and they don't. So this will let you know how many genes are affected and how many mutants are in each of the genes. So a complementation test such as this can reveal mutations in two different genes that can both affect the same phenotype at the end of the day. So here you have a white albino bird from one strain in bird, and then you have the normal coloration, right, in hair, and you can restore the wild type plumage if you cross the two white breeds together. That shows you that, so these are two different white birds, and here is a brown and the wild type plumage. When you cross them, you end up with a wild type plumage, indicating that those two white genes were in different complementation groups, and so when you combine them, you are able to complement and you are able to restore the normal phenotype. Another thing we can do with these type of screens is that we can actually try to knock down a gene of interest, and we can see what it does so that we can see what the possible function of that gene of interest is. So this is done doing something like genetic screens on animal behavior. One of the ways that we can do that is looking at this wild type C. elegans alone, which is in A, and then looking at the mutated gene for a particular gene of interest and seeing that they are doing social feeding in B rather than the individual worms kind of isolated, working separately from each other. Another thing that we can do, so C. elegans are actually very, very cool for genetic screenings because they are easy to manipulate through RNA interference. They can easily be changed up simply by providing them with the E. coli that they eat. You just express the double-stranded RNA of the gene of interest or the genes that you care about in the E. coli, and then feed those E. coli to your C. elegans. The C. elegans will eat them, and they will incorporate that double-stranded RNA into their cells, and they will show their effect. So RNA interference provides a very convenient method for conducting large genome-wide screens even, where you take a bunch, of different double-stranded RNA that could take care of multiple genes all throughout the C. elegans genome, and you put them into E. coli. Each one of these 96-well plate contains a different mutation that you're looking at, different genes maybe, or different mutations at the minimum. And then you put each one of those E. coli colonies on different different C. elegans cultures, and you examine the effect. A wild type culture from one of these mutations or mutants gives rise, a wild type culture is very fertile, it's doing all its things. One particular mutation that you're looking at from here gives rise to C. elegans that are sterile and are unable to mate and unable to produce progeny. And now that's great, right? Theoretically, but sometimes the mutation that you're looking at is lethal or, like we said, unable to produce progeny, and you can study what exactly it does. In that case, you can actually create mutations that are time sensitive or that are temperature sensitive or drug sensitive, so that they are only going to be expressed when they are grown under those particular conditions that they are covered by. So in those cases, you create your mutants under the control of a transcription factor that is either temperature, time, or drug sensitive, and then plate them the same way, and then examine them first in normal condition, normal everyday condition without that agent to trigger the mutation. And then when you switch them to either the higher temperature, lower temperature, to the tetracycline control, whatever it is that triggers the switch, you can examine what happens to those mutated cell colonies and how they grow or do not grow in that response. What exactly happens to those cells as they switch to the mutated form. So here you have the mutagenized cells plated out in a petri dish, grown at room temperature. Why can't I see my thing? And then when you replicate those into multiple plates, you keep one at room temperature and the other one at the higher temperature, you see that some of the colonies disappear because they have those mutations that were not able to survive because of those time-sensitive processes that were essential, and they can no longer do that function. Another genetic analysis that can be performed is called SNP analysis. SNP analysis is looking at single nucleotide polymorphisms. So all along our genome, there are areas that are highly variable that contain these single nucleotide changes at specific points in multiple levels. And those single nucleotide polymorphisms can be used to even fingerprint you, or as well as looking at its likelihood of passing from one parent to another. Now, many times SNP analysis can help you pin down the location of a mutation that causes a genetic disease, because let's say we don't know what gene it is. We have no knowledge of that gene, but there's a particular disorder or a mutation that we're seeing, a genetic disease that is coming up in a particular family. You can, because we have no sequence knowledge or no information or where that gene might be or what that gene might be, it's hard to find. However, you can look for common SNPs and SNP the single nucleotide polymorphisms in that population and see if one of them is kind of inherited more likely in the disease setting versus the normal setting. Once you find that SNP location, once you identify that single nucleotide polymorphism, those are, we would know where it is at that time. And if we know that a particular type of polymorphism is more likely to be inherited along with the disease, it's not because of the actual polymorphism that the disease is happening, but it's likely that it was present close to a gene that is causing the disease. And so we can then look close to that SNP location and look for possible genes in that area and look for which one or if one of them is the result, is cause for the disease of interest. So here you have an example where you have a chromosome pair in heterozygous mother, you have a particular SNP and the recessive mutation, and on the other you have a normal gene and an alternate SNP, which is same as what you also see in the heterozygous father, where again you have the SNP A and the recessive mutation, and because they're very close to each other, they're going to be linked. You have the egg and the sperm form from these individuals, and you have various possibility for how they might be separating. So you can have some which just have the SNP. You can have some that have the A's, the wild types, and the wild type genes, or the mutated genes. This, SNP A and the mutation, SNP A and the mutation. So this one's going to have the disease. Then you have others that do not have the disease, but all of them, they have one copy of A, but they also have B. One copy of B and A, right? So they have the mutation, but they also have the other SNP as well. Here again, you have the B mutation, but then, or not B, the B SNP, but you don't have the mutation because they came with the wild type phenotype. The only time that you see the disease is when the only SNP that you see is A. So this gives you lets you know that wherever the SNP A is on that chromosome, the disease gene is going to be close to it. So recessive mutation in this case is co-inherited with that SNP A. So if we see that in multiple families, we can observe multiple families each time having the same kind of analysis, then we know that the mutation causing disease allele is going to be close to that SNP. And so we can then start walking up the chromosome and around the SNP and see where that gene locus is. So again, here's another example of doing that linkage through SNP. Here you have some healthy controls and some affected individuals, and genes that are going to affect the risk of developing some common disease can often be tracked through linkage to SNPs, because many times these SNPs are going to be variable in the normal versus the affected individuals, and you can then walk up and down the chromosomes to look for mutated genes of interest. Here is an example how you would run this analysis. So there are there are 100,000 locations of SNPs all around our genome. You essentially do a whole genome analysis where you examine all the SNPs and their location and whether they are more or less likely or frequent in people with that particular disorder or not. So you need lots of individuals with the disease and you need lots of healthy controls. And you would notice that if there was a peak for one or more of them, then you know that that is a significant location. So in this case, you're seeing a SNP genome-wide association study. You're looking at strength of correlation, you're looking at SNP location number. So right, these are all the different SNPs that they observed, that they examined. And you look at the correlation. The higher the number, this is the threshold that we are looking at. This would be the significant. And at the end of the day, only one reached the significance. And so we know that this particular SNP is somehow associated with, is more frequent in people that have the age-related macular degeneration or AMD. And so that is basically the 5% probability of false discovery rate, right? So this was the p 0.05. we know that this particular SNP is in the right location, so we can go around that SNP to on that chromosome that it belongs to and see what the gene is that has higher mutation rate or that has specific mutations leading to age-related macular degeneration. Oh, sorry, discard. So hopefully that was clear. If there are any questions about any one of these questions, these slides, or any one of these concepts, please let me know in the chat and I can explain them further in the next lesson. Next lesson is going to be a pure genetics lecture on linkage. So we talked about it a little bit today. We talked about how linkage can happen when there are two genes that are close to each other, that Mendelian segregation ratios are not followed anymore because of their close association, so that they tend to cross over together. So we will talk about when that happens, how does that happen, and how can we calculate if two genes are going to have linkage or not, and how do we calculate the distance between two genes using those linkage ratios. So hopefully that will be up next week. Take care. Bye-bye.